The purpose of the acid-base titration lab was to prepare a hydrochloric acid solution, determine the molarity of another group's unknown hydrochloric acid, HCl, solution by neutralizing it with a 0.5 molar sodium hydroxide or NaOH solution, and monitor the pH during the titration. On day 1, we prepared a 0.5 molar NaOH solution using 2 grams of NaOH pellets and 100 milliliters of water. We did this twice for a total of 200 milliliters to use during the experiment. We also prepared an unknown HCl solution from a 3 molar solution in which we diluted to be 0.75 using 25 milliliters of HCl and 75 milliliters H2O for 100 milliliters total. To determine the unknown molarity HCl solution, in which we had the of summer 7, we slowly dropped NaOH using a burette into 10 milliliters of HCl using phenolphthalein as an indicator until the solution achieved a consistent light pink color, signifying the titration's equivalence point. The moles of NaOH at the equivalence point are equal to the moles of HCl that were initially present since HCl and NaOH react in a 1-to-1 mole ratio. We calculated the volume of NaOH used, recorded the initial and final burette readings for each experiment, and used the formula N1V1 equals M2V2 since they are in a 1-by-1 mole ratio to find the molarity of the unknown HCl solution. The concentration of the HCl solution was determined to be 0.82 molars based on our 2 best, which was within 0.05 milliliters, average data. During day 3, we tracked the pH of the acid solution throughout the titration by recording the pH after each small addition of NaOH, using paper pH indicator from 1 milliliters to 13 milliliters. There was no noticeable change in pH as it remained 1. After this point, we began adding NaOH in 0.5 milliliters increments, at which time lighter red was noticeable on the pH paper. The largest change was observed to be 15.5 milliliters to 16.5 milliliters, with 16 milliliters being closest to 7, showing a pH of 8. After this change, there was little variation in pH between 16.5 milliliters and 19 milliliters. Thanks to these measurements, we were able to determine the approximate location of the equivalency point and create a pH versus volume curve. During the titration lab of pH 7, the pH of pure water is anticipated. Using the graph's midline, we determined that the true pH was approximately 6.75. The results of this lab support the expected behavior of a strong acid strong base titration. A sharp curve, consistent volumes across trials, and minimal variation in calculated molarity. However, several possible sources of errors may have influenced the outcome. These include reading the burette slightly above or below eye level, difficulty judging the exact moment the pink color persisted, small droplets of NaOH remaining on the walls of the burette, or inaccuracies in preparing the NaOH solution. Any of these may affect the recorded volume and lead to slight deviations in molarity. Despite these sources of error, the experiment was successful. The calculated concentration of HCl was reasonable and consistent across trials, demonstrating that titration is an effective and reliable technique for determining the molarity of an unknown acid. This lab reinforced the importance of careful measurement, precise endpoint detection, and repetition to obtain accurate chemical data. Lastly, the process of titration is utilized in many industries, such as pharmaceutical environments, the food industry, and environmental sustainable industries. These examples exemplify the use of titration to manage pH and create substances that benefit society.